This is species distribution modeling in our step 2. So this is the third in our series of videos, and the second video that works through some of the code. And this one relates to the environmental data that you'll be using to predict where species occur in the environment. And again, the work we're doing here is just some examples of things that you can do. Some of this is done for you if you're modeling in the point and click dashboards, but there might be additional steps that you want to take with your environmental data, and then upload those data to the platform. And if you need to do that kind of thing, you probably want to use something like the code that we're going to provide examples to. And again, there should be more examples of these kinds of things coming as the library of code grows within EcoCommons. But once you've fired up your EcoCommons instance, so you've launched a server in EcoCommons and you're in the cloud, you can begin to start to think about environmental data. And again, I want to highlight this paper. I mentioned this at the beginning of each video, but it's a great video or great paper highlighting the process, all the different things that you need to think about when you're doing a species distribution model. Highly recommend that you read this. But today we're going to just talk about some of the code and some of the things to think about when you're looking at your environmental data. So one of the first things that is a problem in species distribution modeling generally is spatial autocorrelation. So you might have spatial autocorrelation in your occurrence data, but oftentimes we have really high spatial autocorrelation in our environmental data. So what is spatial autocorrelation? Well, it just means that this point on the map right here is highly correlated to the point right adjacent to it. So there's not a lot of difference as you go across geographic space in your environmental variables. And that becomes a problem because the model assumes, well, every area that's kind of like this occurrence point is where we expect that species to occur. So it really degrades those fine scale differences that you get from one location to the next or one grid cell to the next. And this is one of the primary reasons that it's probably about half of, maybe a little less than half of the literature on species distribution models has led to predictions that are a little bit over-optimistic. So a lot of times if you have a lot of spatial autocorrelation, you don't have good sampling of the geographic or environmental space that you're working in, a lot of times you're going to get an AUC value that looks really high. And that relates to spatial autocorrelation. And some of the steps that we took in the previous video on occurrence data helps overcome this to a degree. But it's just something to keep in mind and something you might need to account for later in your modeling, especially if your validation indicates that it's a problem. One of the other things to think about with your environmental data, so just like your occurrence records, you don't want really old occurrence records. You want your environmental data, you want the temporal period of your environmental data to match your occurrence records. So ideally, if you're using vegetation greenness as a predictor, and your occurrence records run over 10 years, you might want to take an average value over that 10 years for your greenness index. So you want to make sure that the occurrence records and your environmental data line up. The other thing to think about is oftentimes, if you're trying to make predictions, if you're trying to map out where species occur, a bigger extent is often better. So in our example, we just used Queensland because it's quicker to run. a smaller extent or a smaller set of records. If we had done all of Australia and included the New South Wales and Victorian data, we would have gotten probably better predictions across that larger study area, but it would have just taken longer to run. And then the other thing to keep in mind is the resolution. So the size of each of these grid cells. Smaller is generally better, right? You're going to get better predictions and predictions that are of more use for more purposes if you use a smaller grid cell. And when we come back to this idea of extent, so a larger extent is better, but it's also better if you train your model on areas where you know the species is going to occur. So if these cells that have been masked out here are water for a desert species, so you know that each one of these cells has over 80% water in it. So it's unlikely that your species is there, let's say. You might mask those out, because if you don't mask those out, if you include those places where the species, you know the species cannot occur, but you include them in your model, you're going to get an inflated AUC value. It's going to look like your model's doing better than it's actually doing, because it's discriminating between those places you know it can't possibly occur and the places where you have records. It's much better for the model to work hard on the areas where you're not sure. It could be there, it might not be there. So the more of the areas where you're sure it's not, you can mask out of the model, the more realistic your evaluation statistics will be. They'll reflect how well did we do predicting into areas where we weren't sure if it was there or not, as opposed to, I know my rainforest species isn't in the desert. So masks can be useful. They're not widely used, but certainly it makes your model work on the areas where you're not sure, instead of including areas where you're sure where the species doesn't occur. So getting back to this idea of resolution. So if a smaller grid cell size is better, well, what do you do when patch size is really important? So my species requires a huge area of trees, you know, thousands of square kilometers of forest for it to be found there, let's say. But I'm only modeling at, you know, a 250 meter grid cell resolution. So my cells are only 250 meters by 250 meters. Well, what you can do is you can combine, so you can keep your cell sizes small, but use something that's called a neighborhood function to capture that larger scale activity. So for example, if the neighborhood cell that we're going to calculate a value for is this black cell, but we're going to calculate the value of that black cell from cells surrounding that cell, right? So for two cells in any direction, we're going to calculate, say, the sum of the percent of forest cover in all of these cells. If it's all 100%, then you've got a really big value here when you're in the center of the forest. As you go towards the edge, it gets smaller. If you're in an isolated patch of forest, it gets smaller again. So it's a good surrogate of patch size. It's a good surrogate of edge effects and of connectivity. So it's not a bad way to capture a lot of things with a proxy of all those things using a neighborhood function. So that's one way to handle these kinds of things. And we'll go through some code that shows you how to do that. It's fairly simple to do. And when we're thinking of our environmental variables, you're going to create much better predictions that will stand up over time, and they will stand up across new areas where you don't have any records. That's going to happen if the environmental variables that you're using are highly related to the requirements of your species. So oftentimes we're just using temperature and rainfall and things that kind of are proxies for, you know, the rainfall might relate to the abundance of food or, you know, different things, right? You can argue that most of these variables are proxies of the actual variable that's important to your species. But the less you're using proxies and more you're using those direct variables that are important to your species. So the amount of spinifex vegetation in this patch of mallee, for example, or the amount of dead snags in this forest, right? Those are great variables to use. Unfortunately, a lot of times we don't have data on those more fine scale habitat variables or structure variables that are important for your species. So we do end up trying to get proxies that are as close to your species' ecological needs as possible. And the more of them that you use, the better, that are related to your species. You really run the risk of overfitting a model if you're using variables that aren't related to your species, and then when you go to predict somewhere else, your model fails miserably. Right, so the more that it's one of the key messages of this module is the more related your environmental variables are, the better for predicting species distributions. It's also worth considering how these environmental data are often generated. So, you know, we're just putting a grid across Australia, right? And within each grid cell, we're saying, well, how much rainfall was there? Well, there's no grid cells here. There's no rainfall stations actually giving us data here in the middle of Australia. So a lot of work, you know, by a lot of researchers goes into making the best possible estimate of what values are in these parts of the grid. But just keep that in mind, that if you're trying to model something in the middle of Australia, there's a lot of uncertainty with what those actual environmental variables, what their values actually are. And so if we look at how we deal with that kind of thing, if we've got a bunch of temperature stations that are collecting temperature data across our study area, we're just going to interpolate, right? We're going to use a function that says, well, it's somewhere between, here we have 24, so maybe this one's 23. You know, you're just going to take an estimate of, based on the values you do know, what are the values that you don't know? And spatial interpolation, especially with these kinds of things, is a science unto itself. So keep in mind how your environmental data was generated, how it was interpolated, and how you should be using it when you're doing these kinds of models. So in this module, we're just going to cover basically first the first step that you need to take when using most of the algorithms that are available in R, when you're using Maxent, is all your environmental data needs to match. You need to be able to stack them on top of one another and for the grid cells to line up perfectly. So that means the grid cells have to be the same size in each environmental layer. So that's called resolution. You need to make sure the extent is the same in each one of those. That's the outline of your study area. It needs to match in each one of those environmental predictors. And you need to have the same coordinate reference system. And this is where a lot of people go awry. And if you're not careful, you know, you can take two environmental data sets and stack them, but they have different coordinate reference systems, which means that when you overlay your occurrence points for that species at that location, it's picking up the correct location for one environmental variable, but it's picking up the wrong data, the wrong environmental data from another environmental data. or another environmental raster layer. So there's a lot of work to be done to make sure that your raster layers line up. We're also going to run through a focal function. Again, it's fairly simple to do. It's just something you need to think about when you're doing it. We ran across reclassifying data that can be handy for some data sets. We'll talk about that, but we had an example in the previous video of how to reclassify raster data. And finally, we're just going to show you how to stack these environmental variables. And the reason that's so handy is if you have a stack of environmental variables, and each raster in that stack has exactly the same names as the names you used in the variables to build your model, you can predict to the whole stack. So that means you can predict across all space, or if you go forward in time, you need to give your stack, you need to give the rasters in that stack the same names, but you can predict into the future, right? Or back in time, whatever you want to do. You can predict across an entire, across all of Australia, even though your study area might have been northern Queensland, by using raster data that have the same names and have the same, and you can stack them because they all have the same resolution, CRS, and extent. So there's not a lot to it, but it is a process. And again, the point and click dashboards available on EcoCommons take care of this for you. But if you want to do something with that data, so if you want to download it and reclassify it and then upload it again, you need to keep these things in mind. Or if you just want to upload your own raster data or use it in the R environment. Either way, these are good things to know. So first, we supply a data, a folder in the environments online. So online you can download all the scripts, and there's also folders with data in them. And if you put those in your working directory, you'll be able to upload data. So the last video highlighted how to set your working directory. So if you need to watch that, that's in step 1 at the beginning of that video. But you need to set your working directory. And within your working directory, you'll have this folder called raw data and this file with this name in it. And you can grab that off our website, both the folder and the file. And this is just an example of how to bring a file into your local environment and make it a raster. So it's just reading it in, basically, reading in your TIFF file. And let's see, I'm assuming that most people who are watching this have accessed the code and have already run through the code. So I'm just highlighting what some parts of the code does. And I'd encourage you to use these little number signs and put comments in where things that you'd like to remember, where you want to add more stuff than we've already added as comments. So I'd encourage you to have run these already, but either way, I'd encourage you to open up the script and take notes as we go through this stuff. But once we've uploaded that raster into our local environment, we can then change the coordinate reference system and the resolution to match that of base by simply using this command project raster, my new raster, and base, which is that old raster that is the same extent that we want to set everything to, same extent, resolution, and coordinate reference system. So this does this automatically. There's commands within this function that you can use to fine tune how you do this, and there's other ways to do this. So if this step is particularly important, how we're warping a raster or how we're resampling data to make it finer resolution, those kinds of things. If they're of particular importance in your field, there's a whole lot more you can do. This is just taking the default values of what usually works pretty well. So this project raster does that all for you. You then still need to run this mask command over your new raster AWAP2. And AWAP, by the way, is an index of soil wetness. So that's what this is referring to. But anyway, you still need to mask out those areas that are outside the extent of your study area, because this returns a grid that's a little bit bigger than our study area in the base. So you still need to run mask. Once you've done that, you can rename this just as AWAP. So that just means this is a copy. AWAP is the same thing as AWAP3. Let's see, I think I should have shown this a little bit earlier, but that base is just loading the base in if you haven't done that already, so that should have been done up here. But that's how you do it. That's how you load something from your local directory. And you need to set the CRS of base, and otherwise this function won't work. And then we're also looking at another data set. So we're looking at this data set called NVIS, which is a nominal or a categorical variable where each number in the grid corresponds to a class of vegetation. So it's not a continuous variable, it's a categorical variable, and it's of vegetation classes. So we're just going to take a look at that in the R script. And when we did that, actually, we found that there were no freshwater wetland variables, which would have been the most valuable one to predict where frogs occur in the landscape. And there were none of the other vegetation classification variables related to where our frog occurs. So we looked at it, but we didn't use it. So you can see that in the script. We took a look at it, but it wasn't something that we thought would be useful, ecologically relevant to our species. Perhaps we could have reclassed some of those vegetation classes. There's a lot of vegetation classes. We could have reclassed those into forest and non-forest, or you can reclassify any of those categorical variables into a broader category. And again, the way we do reclassification was covered in that first video. But again, for our purposes, we didn't think that things like open eucalypt woodland was a useful classification for figuring out where frogs are in the landscape, or any of the other classifications that are in that data. You might disagree, and you might want to reclassify some of these things so that you get a categorical variable that's useful to you. So we also we upload a raster of wetland cover, and we show how to create that kind of a raster. So when you have categorical data, say you have categorical polygons, you can use the Rasterize function to produce a result where the percentage of cover in each one of those grid cells is reported. So if half of your grid cell is covered by wetland, for example, in this example, you get a value of 50% or 0.5 in your wetland cover variable. So it's a good way to, if you want to turn your categorical variable into continuous data, one good way to do that is to calculate the amount of cover that's found within each grid cell from the original source polygon layer. So anyway, that's what's been done here. We set the CRS for that species, or for that, I'm sorry, for that raster layer. We then use project raster, and we mask that. So those are the kinds of steps that you need to think about for each raster layer that you, each environmental data raster layer that you produce. You want them all to match base. And then here's an example of a focal function. So that's that neighborhood function. And what we're going to do is we're going to sum all of the, so we have a percent cover, right, in wetland cover. So wetland 3, it gives us the percent cover of wetland in each grid cell. And then we're going to sum all of the variables within, so a grid 11 by 11 around that central grid cell. So you've got to have an odd number here because that is going to include the grid cell that you're using in, and it's going to return in that focal grid cell, it's going to return the sum of all the percent cover of wetlands within basically five grid cells in any direction, right? So a total of 11 by 11 grid laid over the top of your focal grid cell, and it's going to sum all those values. So if there's a lot of wetlands around, a lot of wetland cover, you're going to get a really big number. If there's very little wetland around, you're going to get a smaller number. And then here we demonstrate that you can download data directly from online. So you can do this in EcoCommons. When you go to the environmental data and you're looking through environmental data, if you click on view the details, there'll be a web address where you can download that data, and you can simply go EVI and then call raster and type in that web address, and it will upload the data from the web into your local environment. So that's a handy trick. And again, we're going to set the CRS and the extent the same as the base. So EVI is a greenness index, and we're going to mask it, and then we're going to rename it just EVI because that's simpler, right? And the reason I tend to go 1, 2, 3 on object names is if I go wrong somewhere in my script, I don't necessarily have to rewrite everything. I just have to rewrite the line where it went wrong and then run it from there. So it's just a habit I have in coding, but it's not one you have to follow necessarily. So this is just doing the same thing for our bioclim variable. Bioclim variable 5 is it's downloading that from the web, and it's setting the extent or the resolution and the CRS as the same, setting the extent as the same, and giving it a new name. And here we're writing it to a folder called predictors in our working directory. So that's important because we're going to predict to these variables later, because we want to predict across not just where we took the data, but across our whole study area. So in order to do that, we need to save an example of each one of these files. So we did that for wetland connectivity, we did that for EVI. So this is just an example of what's repeated throughout the script, is you want to write these files to your predictors file. And then we do the same we just repeat those steps for bioclim 6, for bioclim 12. So those are, going from temperature variables to rainfall related variables. And then we're going to stack. These are all our current data, right? So the bioclim data that we downloaded was based on current environmental conditions. So that's usually averaged over the course of the last decade or two, usually. And that average represents your average temperature or average rainfall, right? Whatever the 6 or 5 or 12 refer to. And when we stack those, if our extents line up, if our CRSs line up, if our resolutions line up, we should be able to stack them. And when they're stacked, you can use the stack as one object when you're running your models or when you're predicting. So it's a handy way to deal with all of your predictor variables. And here we're just going to make sure that their names match. So a lot of times when you do this, when you stack a bunch of variables, it might not return the same names. So you just want to double check that. And if you need to, you want to reset the names of your raster stack to match those variable names exactly. So it's an important step. And then you want to repeat this again with future climate data. So we've chosen here, we've found some future climate data, and we want to predict into that. Like we want to build a model based on current conditions, and we want to predict into future conditions. So we found on EcoCommons an environmental data set which uses the same kind of model to generate its predictions as our current data used. And so we've used WorldClim compatible current climate data with WorldClim compatible future climate data. And we're using the same variables. So we're finding bioclim 5, but that's predicted under an emissions scenario of, RCP 2.6 or whatever it might be, right? So that's the emissions scenario, and we're using a climate model that uses the WorldClim sort of modeling framework. So we found that variable, and if you have questions about how to do that step, you can see the introduction, the first video in this series. But we're just downloading each one of those variables, loading it into our local environment from the website, and then we're writing that to a new folder called Predictors Future. So in Predictors Future, we've used different data for the bioclim variables, but we're giving them the same name as the names that we used in our current data folder. So the file name will be the same. The raster for bioclim 5 will have the same name, but the predictors future folder contains those future data. So, and then we'll need to make sure that we also copy across the AWAP raster, the wetland connectivity, and the vegetation greenness rasters into that future data set. So those, we don't have future predictions of soil wetness. We don't have future predictions of wetland connectivity. We don't have future predictions of vegetation greenness. So we're going to just use the current data that we have and put it into the future folder. It's been shown that generally you get better results, even though we're making assumptions that these things are static over the next 50 years or however long you're modeling into the future. You still often get better results than if you're just using kind of bioclim variables. Not always, it depends on your question. The important thing is that each folder that you're going to predict into needs to have exactly the same raster names within it. And when you stack those and use them in your model, they need to have exactly the same raster names as you've written into that folder. So those are the key parts of making things line up. And again, this kind of wrangling of data is a real hassle. And the great thing about the point and click environment in EcoCommons is it does all this for you. If the data is already in one of those selected databases or data sets that's available at the click of a button within EcoCommons, you can take care of all this automatically. But oftentimes you want to maybe reclassify one of your rasters, you want to use a focal function, you want to bring in different data that's not available on our platform. You need to be across these kinds of steps in order for it to work. So once you stack the data, you know, you can plot that stack and you get, and this is an example of without the new names attached to it. So this is the names that came out by default. So that's why we had to rename this raster stack, but you can just press plot and the name of the raster stack and you'll get, this is the kind of output you'll get in R. So an example of, in our case, all 6 of the environmental variables that we're using in this data set. Anyway, that's a quick summary of how you need to format environmental data and some of the things that you need to think about. We'd encourage you to, you know, if you run through the code and it's not making much sense, and it's because you don't have a good understanding of the basics of coding in R yet, I'd encourage you to visit this website or visit the EcoCommons website where there's some videos that Amelia's done that take you through those steps of you know, how coding works in R generally, what are objects, what are loops, all those kinds of things. We also have use cases that will be available on the website, so you can check out this ecocommons.org.au. You can also, if you want to visit the EcoCommons website before it launches in November of 2022, you can contact us and we'll probably give you access to the platform. Just keep in mind that it's not going to be as reliable as it will be after November 2022. And if you do use it, please give us feedback. There's a little feedback button on the side if you do happen to use it. And if you're interested in what's coming in EcoCommons, again, I'm really hopeful that EcoCommons over the years is going to become a place where people can share scientific workflows. And we're just working through some examples, but we're hoping that more examples will become available down the track. And there's a lot of workflows that have already been very well developed within the EcoCommons platform. We just are looking for more in the future. If you're curious about what's coming, generalized dissimilarity modeling is coming soon, risk mapping is coming soon. Visit our website and sign up for our newsletter, and you can keep up to date with where we're tracking on different workflows. And none of this is possible without all the help of our partners and our collaborators. And so we just acknowledge their contribution to make what we're doing possible. And thank you for listening. That wraps up the environmental data module. And the next module is fitting your model. So we'll work through a couple common models that you might want to fit for a species distribution model, how you do that in R.